So today we're speaking with Associate Professor Kate Nautiyal, who's in the Department of Psychological and Brain Sciences at Dartmouth in New Hampshire.
Great.
And Kate's doing some really excellent work looking at, particular focus on one of the lesser known or lesser studied sort of 5-HT subtypes that's of relevance to the action of psychedelics, and that's 5-HT1B. So I was wondering, Kate, if you could tell us a little bit about your sort of background, your research background, how you got interested in the 5-HT system, and what are the primary research questions in your lab at the moment?
Sure. Yeah. So I'm a behavioral neuroscientist, and I'm really interested in understanding how the brain modulates different reward-related systems and behaviors. So I'm really interested in the pharmacology of disorders of the reward system, including depressive, anxiety, impulsivity, substance use disorders. Yeah, I'm really interested in that sort of big black box between the really molecular ions like biochemistry signals to the really big behaviors like why do we feel sad? My work in my lab really targets this black box area. So we use preclinical mouse models. And this way we can dissect the different circuits going on and make different manipulations pharmacologically and genetically and look to see the resulting behavior. And my work, the focus of my work has really been on serotonin because of its obvious implications in depressive and reward related behaviors. But it's really been understudied, right? When we think about reward, we think generally about a molecule called dopamine. And I think the serotonin has really been underappreciated in this area. And it actually has 14 different receptors. So it
Yeah,
has
yeah.
a lot of different ways it can signal. And I think the idea of psychedelics and classic serotonergic psychedelics impacting this system just got really interesting to me and trying to figure out how serotonergic psychedelics are having these persisting behavioral effects. What are they changing in the brain to cause these long-lasting behavioral effects? So
Yeah,
that
yeah.
was kind of a long-winded way of
No, no,
how I
it's
got
great.
interested in serotonin.
No, it's great. Yeah, yeah. I think a lot of people are probably not aware of all of the serotonin subtypes. So just to set the scene, we have these 14 now known 14 different subtypes. They're all GPCRs apart from 5-HT3, which is an ion channel. I think people who are studying psychedelics and 5-HT signaling are very familiar with 5-HT2A, 5-HT2C, 5-HT1A. So 2A is this GQ coupled excitatory, largely sort of postsynaptic receptor, right? But 5-HT1B, I feel like is much lesser studied in this context. So could you give us a bit of a profile of sort of 1B and how it differs from 2A? Because it is quite different.
Yeah. And I think people are familiar with 2A because of the really strong work implicating the 2A receptor and these acute subjective hallucinatory effects.
Just one point quickly is that actually, compared to even things like 5-HT6 and 7, the interaction with psilocin, the active metabolite of psilocybin is actually more prominent with 5-HT1B, right? Although no one's really studied it until I came across your work.
Exactly. Yeah, I mean, I think the jury's still out of what it's doing, right? Because a lot of this data comes from receptor binding, but then as people like Brian Roth say, you really have to look at like functional effects. So I think we're still, in the midst of trying to figure out what receptors psilocin can have functional effects at and what it's acting through. And so, yeah,
Right.
that's exactly what our work is sort of targeting functionally in vivo, looking to see how non-2A receptors might be involved in these persisting behavioral effects of a single high dose of psilocybin.
So what's a quick profile of 1B,
Yeah,
in terms of location and function and what we know
yeah.
about it?
So 1B is so complicated. I think that's why I'm so interested in it. It's really located throughout the brain, but on the presynaptic nerve terminals.
Yeah.
So whereas the majority of serotonin receptors and other receptors are located sort of on the cell body and the dendrites, known as like the inputs of the neurons. 1B, the serotonin 1B receptor is located on the end terminals, regulating the output or the release of the different neurotransmitters. And so it's an inhibitory receptor.
Mm.
So binding of the receptor inhibits the release of these neurotransmitters. And it does so by acting both on serotonin neurons and non-serotonin neurons. So it's quite complex also in being able to figure out where it's having its effects because of this sort of distal effects on the output. It's also located widely throughout the brain enriched in like basal ganglia reward related regions and frontal regions and hippocampal regions. So it's really has the potential to cause modulation of a large number of neural circuits. And yeah, and we've seen its impacts on reward related behavior,
Mm.
including depressive phenotypes, impulsive phenotypes, aggressive phenotypes.
Yeah, I
And
recall seeing some of these compounds when I was looking for, as a chemist, I'm always fairly chemistry centric on a lot of the research that's been done, and you can find some interesting sort of fairly advanced probes and other compounds that were being developed preclinically. And a lot of the research is really focused on aggression and reward related behaviors, so self-administration of cocaine and other things, which I thought was kind of interesting. So
yeah,
yeah, I think I learned at an ACMP conference a few years ago when I first met you that you were actually involved in developing the 5-HT1B knockout mouse line, right? Yeah,
yeah. So, well, my postdoc advisor made the original 1B
right.
knockout line in the like late 1980s
Cool.
when knockout mice were even first just being developed. And
Yeah.
he claims it was the first brain relevant or neuroscience relevant knockout mouse.
Oh, that's super cool.
Yeah, and incidentally, I always asked him like why he chose this receptor, and he said, well, it was actually convenience because it only had one exon. So they didn't have to
Yeah.
worry about which exon to disrupt. They could just do one shot. And so it was out of convenience, but then they were able to find, really profound effects on impulsivity and aggression. And then 20 years later, I came into his lab and we still hadn't worked out if these phenotypes were developmental or not, right? Because
Mhm.
these knockouts were lifelong. And we hadn't figured out the circuitry through which these effects of serotonin 1B were acting to influence behavior. And so we
Right.
came in and made this tissue specific and inducible knockout that has been really helpful in trying to figure out where and when the 1B receptor is impacting these behaviors. And then also most recently to figure out how psilocybin or how it's influencing the response to psilocybin, I guess.
Yeah, awesome. Yeah, yeah, I really loved the molecular psychiatry paper. I think I first saw it as a preprint, and I was like, this is pretty neat because I don't think, as far as I was aware, I don't think anyone had really looked at 1B, and it's a fairly well defined but understudied interaction of psilocin within the 5-HT system. So for those who are not familiar with the paper, maybe, could you give us sort of some of the main sort of research approach and what some of the main findings were? Because there's a lot of really interesting data in there.
Sure, yeah. I mean, I think the first challenge was to come up with like robust and reliable and reproducible preclinical behavioral effects. And I think that's
An,
still a bit
an
of a
ongoing
challenge.
challenge for psychedelics and acute behaviors.
Yes,
Yeah.
it's still an ongoing challenge. And actually, we have a paper hopefully forthcoming soon, really dissecting out all these different effects, all the different variables that can affect the post-acute behavioral effects of psilocybin. The graduate student ran like 700 mice looking at all these different variables,
Amazing.
trying to pull out like what is most influential in getting these really big antidepressant and anxiolytic effects. So I think
Super
that
cool.
was our first challenge. And once she found a protocol and paradigm that we felt was strong enough to be able to dissect, so she injected psilocybin into both control and knockout mice, so mice lacking the serotonin 1B
Yeah.
receptor, and then looked, 1, 2, 3 days later at their behavioral effects. So she looked at anhedonia, right? So how pleasurable they find intake of sucrose, looked at some anxiety-like behaviors in the elevated plus maze, right? So this is how much time they spend in a closed cave-like safe arm or open anxiogenic cliff-like arm, and basically found that while psilocybin decreased anxiety-like behavior and increased hedonic responding, in the absence of the 1B receptor, you see attenuated effects. So you
Right.
see that psilocybin doesn't have as big an effect on reducing anxiety and doesn't increase sucrose consumption as much. And then
Super
she also
interesting.
followed up with some pharmacology. Basically, instead of using the knockout, just administering a receptor antagonist
Mhm.
to 1B along with psilocybin. And this showed us that it probably was because psilocybin is acting on, or psilocin is acting on the 1B receptors during this acute period to have the effects on behavior. And then she also did some neural work. basically looking at how psilocybin changes the neural activity patterns in the brain, and then looked to see how the absence of 1B was affecting that.
Right. Yeah. I think I saw some sort of CFOS, like immediate early gene expression changes in the
Yeah.
paper, right? What was the sort of top line result from that work?
Yeah, there's not a clear answer. I
Okay.
would say there are some brain regions in which 1B has no effect on the neural activation, like the claustrum, for example, which makes sense because that's a brain region high in 2A and not really in 1B. And so we see equivalent levels of activity following psilocybin in those brain regions. Other brain regions, like some of the striatal and amygdala regions, we do see quite an effect of 1B knockout on these brain regions. So I think, and she also did some network analysis to try to understand the connectivity and how synchronized or desynchronized brain regions are. So I think it's a whole big nut that we have yet to crack, but I think it's further complicated by the fact that we knocked 1B out of the whole brain.
Yeah.
And so now she's going and just knocking 1B using the conditional knockout out of
Mm.
specific cell types and brain regions, so we have a better ability to understand
Yeah.
what circuits 1B is affecting.
Yeah, it's amazing to have access to all these like modern neuroscience tools for doing conditional sort of region-specific knockouts with
Yeah,
psychedelics in the modern age. It's
yeah.
super cool. Just because I know some people will be curious about it, and it's something that I have been grappling with for the past few years in terms of understanding the underlying and fundamental mechanisms. But what are the contributions, based on your paper, what are the contributions of 1B to acute effects like head twitch response, a sort of proxy for human psychedelic activity potentially, and other sort of locomotor behaviors and other things?
Yeah, yeah. So we don't see effects of 1B on head twitch responding.
Right.
And that sort of checks out from some older work from Mark Geyer's lab.
Mhm.
We might see some effects of 1B on the locomotion. So we give a high
Interesting.
dose of psilocybin that causes hypolocomotion in controls, and we see some attenuation. It wasn't significant really in all cases. It
Okay.
definitely is reproducible. So I think something is there, just not sure through what circuits and which receptors. In subsequent studies, knocking 1B out of, say, just the cortex, we don't see this attenuation of hypolocomotion. So
Interesting.
perhaps if there is an effect, maybe it's through some subcortical basal ganglia type
Right.
effects.
Yeah.
The effect isn't very large, but I do think that there's something there through which 1B is influencing locomotion. But yeah, head twitch we have not seen at all impacted by 1B.
Yeah, super interesting. Yeah, well, congrats again. It's an amazing
Thanks.
paper. And yeah, as you keep referring to the grad student working on it, I do note that it's just the two of you on the paper in molecular psychiatry, which is a pretty impressive feat given
Oh,
how much
thanks.
really interesting fundamental data is there.
Yeah, 16 has done so much work in trying to figure this out and really careful work trying to come up
Mm.
with these animal models. I think the paper really showcases her hard work. Yeah.
Yeah, awesome. Well, thanks so much for your time, Kate. I really
Yeah.
appreciate it.
Thank you.
Yeah, really looking forward to the future publications from your lab.
Yeah. Thank you. Yeah.